In Demo 2, I'm going to go to a region of the genome and have a look at the structural variants there. So I could have, from the ZAP70 gene page, just clicked on that location tab. But coming back to the main page, I just wanted to show you how to get there if we weren't already in the Ensembl browser. I'm going to do a very similar search as I did for Demo 1. But you can click from the search results directly on the location. So now I'm in the region in detail. This is actually a 1 megabase or smaller region of the genome. So I've got ZAP70 in the middle and neighboring genes on either side. For example, the TMEM131 gene, the ANKRD36B gene, etc. And now if I scroll down, I can see in much more detail the region for the ZAP70 transcript. So just to briefly let you know what tracks are on by default, I have uniprot proteins aligned to the genome. So the regions of alignment are there in yellow solid boxes. And those are just letting you know where the exons fall, basically. And then you've got human reference sequence and ENA, cDNA, and mRNAs also aligned to the genome. I know that because I can go over the track and go over I. So if I hover over those I icons, then you can see a little description about what that track shows you. I'm going to scroll down a bit more. You've got a legend at the very bottom that lets you know, for example, that the blue transcripts here are processed transcripts or non-coding transcripts. And then you have protein coding transcripts as well in red and gold. But I'm going to now add structural variations, and you might remember from Demo 1 that to add things to the display, you can configure this page. So I've got a lot of different tracks, over 100 different tracks I can draw. I'm going to scroll down to structural variants on the left. And I'm just going to turn on all the sources. Okay, you could also turn on, for example, decipher. That DAS means that it's an external source that Ensembl does not maintain. I'm going to change this to normal, and then save and close the menu. So now if I scroll down, I have 2 decipher tracks in this location, and you can click on them for more information. And I also have structural variants that Ensembl has in the database. So if I click on it, I can see this one comes from DGVA. However, what if we want to look at more than 1 megabase? At the moment, I'm looking at around 26 KB. I could zoom out all the way, but in this region and detail view, I can't get over more than 1 megabase. So for that, I can go to the region overview page. So you might be able to see we're in region and detail. I'm now going to click on region overview. Now this is going to give me the potential to zoom out a lot. So right now I'm zoomed into the ZAP70 gene. I'm just going to zoom out further. Okay, so now I'm in a 500 KB region, and you can see the structural variants there.